So, hello, welcome to today's bite-sized bioinformatics. Today I'm going to be talking about exploring the NC3R's experimental design tool for robust and reliable experiments. So I'm going to spend most of the time actually on the tool and showing you how to use it, but I thought it might be useful as an introduction to just go through a couple of concepts of experimental design that you will come across in the tool to hopefully give a bit of background on that. So the first thing that is a really important part of thinking about experimental design and is mentioned quite a few times in the tool is about bias. So bias is basically any kind of systematic error that can affect the results of a study, and that can be either intentional or unintentional. Often it is unintentional. I don't think many people intentionally induce bias, but it's something that we want to be aware of and take into account when we are designing our experiments. And there have been studies done on the impact of bias, and this quote here says that the lack of bias-reducing measures such as randomization and blinding, which I'll go into more detail in a second, can contribute as much as 30 to 45% inflation of effect size. So this can have a real impact on both the size of the results, but also whether your results are positive or negative. So as mentioned a second ago, randomization and blinding are two really important factors that can help us to reduce bias in our studies. And studies that do not randomize and or blind in their study designs have more false positives generally. So randomization, as you might expect, is randomly assigning our samples into our different groups. And we can randomize either just overall into our groups, or we can also randomize within things called blocking factors. And this helps us take into account any nuisance variables or confounding factors that might also have an impact on our factor of interest. So some examples of these are things like times of day of the experiment, litter or cage if you're looking at animal studies, the person carrying out the experiment, things like sex, age, body weight, or some other measure that is related to our outcome. So starting cell numbers or size of tumor, if you're looking at tumors, for example. To randomize, we always want to use a random number generator or things like flipping a coin or rolling a dice, or as you will see later, using the experimental design assistant. We don't want to just rely on human randomness, as we actually aren't very random, or not as random as we'd expect. So the other thing I mentioned is blinding. This is where the person who is carrying out the experiment does not know which group necessarily the animals or samples that we're working with belong to. And we can think about this at multiple stages in our experiment. So we can think about it when we're allocating our groups, when we're measuring our outcomes, which is a really important one, particularly if we are doing sort of slightly more subjective measurements. So if, for example, we're counting cells, then we can quite easily bias the results if we know which group the measurement that we're taking belongs to. Also, generally, when doing the experiment and also when doing the analysis, we can think about blinding as well to try and reduce our bias. So the other thing that's quite a key concept that comes up in the experimental design tool are about experimental units. And these are really important to identify before you start your study. They're basically equivalent to our biological replicates within it. And so it's essentially what you want to make population inferences about. Generally, if you're doing an animal study, it's individual animals. It can be a cage of animals, though, or a litter, or a part of an animal, or cell lines, or whatever it is that you're looking at. And I'm going to go through a few examples as well. Another way of thinking about it is the unit that can randomly be assigned to an intervention independently of all other units. And hopefully these few examples will make that slightly clearer as well. So I'm going to use animal-based examples because I think they're sort of easiest to visualize. So we might have these 4 mice here. If the treatment that we're giving is in the form of an injection, for example, we can take these 4 mice and randomly assign them into either getting a control or a treatment, for example. And which group this mouse gets assigned to does not then impact any of the other mice necessarily. If, on the other hand, we have 4 mice in 2 different cages, so 8 mice total, and we're giving them some treatment or intervention in, for example, the water that is being supplied to the cage, we can't separate out 2 mice from this group and give them a different treatment to the other 2 mice. And so therefore, in this case, the whole cage would be the experimental unit rather than the individual mice. This can also be the case if, for example, you're treating a pregnant mother. If you're treating the mother, then all of their litter will not be able to be independently assigned to a different intervention. And so therefore, the experimental unit in this case would be the whole litter. If, on the other hand, you were treating the individual pups, then each individual pup would be the experimental unit. So that's just a very brief introduction to a couple of concepts when it comes to experimental design that will hopefully come in useful when we go through the tool as well. There are also other guidelines and tools that you can use when thinking about designing your studies and reporting your studies as well. So the Experimental Design Assistant has sort of been derived from the ARRIVE guidelines. So I thought these would be useful to highlight as well. And these are they're actually reporting guidelines, but they're really useful to think about at every stage to make sure that you've thought about things that you will want to report at the end, right at the start of the study. And this basically provides a checklist of recommendations ranging from study design, sample size, all the way down to your sort of results and statistical methods and things like that. The other guidelines that are really useful when you're thinking about planning studies is the PREPARE guidelines, and these are more focused on planning before you carry out your studies, but these are much more broad, much more wide-reaching. So they also include things like literature searches to make sure that you've properly formulated your question and looked at alternatives, your kind of ethical issues, legal issues, and a whole host of things like making sure that you're using the right facility and things like that. So just to point out, the tool is in a whole sort of ecosystem of guidelines and things that can help you to design your experiments as well. There is also the recently developed ARRIVE study plan, which is very much complementary to the design tool as well. So it can be useful to have a look at that as well. So now on to the actual Experimental Design Assistant, or EDA. It was developed by the National Centre for the Replacement, Refinement and Reduction of Animals in Research, or NC3R's, which is much easier to say. And it was developed to help achieve their goal of making sure that people are really following the 3 R's when it comes to animal research. So for those of you who aren't familiar, these basically replacement aims to replace as much as possible the use of animals in research. Refinement wants to make sure that you are using the most refined methods, so you're causing the least harm for the most benefit, basically. And reduction is using the least amount of animals possible to still achieve your scientific goal. So the EDA has been designed to help guide the design of experiments. As you might have picked up from the fact it was developed by the NC3R's, it is mostly focused on animal experiments, but I think it can also be applied more widely as well, because it helps to support and provides advice on things like sample sizes, our bias reduction, statistical analysis methods as well, and helps us to think about any confounding factors and things that we might have as well. And also at the end, it can produce a really nice report and a diagram for your experiment to help with visualization that you can share with other people, that will fill a lot of the criteria that you need for animal experiments, to show that you've thought about your 3Rs, but they can also be helpful, as I say, for other types of experiments too. So this is a sort of high-level overview of the process that we might use when using the EDA. So firstly, we will start off drawing our diagram. We'll then fill in information in our diagram, and we can use some tools within the EDA to critique it, which means it will give us suggestions on how to improve both our diagram and our experimental design itself. It will also help us to choose an analysis method and calculate the sample size depending on how complex the experiment is. And you can also do things like generating a randomization sequence, so you don't have to flip a coin to do that or anything. And then we can share and export our final diagrams. We can also, after we've carried out our experiment, update some parts of the diagram, which will then give us everything that we need for reporting so we can follow the ARRIVE guidelines that I mentioned earlier. So that's a very quick introduction. I thought probably the most useful thing for this session is to actually just go onto the website and have a play around with it so you can see how it works. So I will get that opened up now. So this is the first page you'll get when you go on the Experimental Design Assistant website. To use the Design Assistant, you will need to create an account, but that is free, so hopefully not too onerous. Won't put too many people off. But I thought I'd also just highlight a few more things on the website before I go into building my diagram. So you can see it's a free resource. It will remain free, I would imagine, because it's by the NC3R's. It's been used by over 20,000 researchers worldwide, apparently. So it does get used, and it is helpful, I think, once you know how it works. There is also a whole lot of information about experimental design here. So if you're not very comfortable with some of the different aspects within it, you can find out more here. It gives general information on things like defining hypotheses, understanding experiments, and it also has a fairly helpful glossary. So if when you're creating your diagram you forget what something means, you can come here and it has a lot of definitions for the different things that are covered as well. I'm not going into too much detail on all of the specifics of experimental design because we don't have a whole load of time. So I will go back to this page and show you how we might go about building our diagram. So as I say, you need an account, so I'll sign in here. Once we've signed in, you get this dashboard. You see that they do live demonstrations of this as well, the NC3R's. So if you want more detail on this, you can sign up to one of those as well. But hopefully this will be a good introduction. It also shows any previous experiments that you've created, and you can see that I've got a few here. And you have the option to either create a new diagram or to get started with a template. And so this is something that's really helpful, I think, is to have a look at the templates here. So when they created the EDA, they've also created 5 different templates of different study designs that you might need, ranging from a fairly simple 2-group comparison, where you have individual animals as experimental units and you're just doing complete randomization between 2 groups. And underneath it also provides an example of the kind of study that you might use in this or that you might be using the design for in this instance. So for example, looking at the effect of a drug compared to a control on plasma glucose levels. It also has a whole load of other templates. So using crossover, where you are using the same animal and randomizing between two different interventions and then testing something and then doing the opposite intervention and testing again. There's also more complicated ones, so where you have multiple different animal characteristics. So in this case, they're looking at, for example, wild type males, knockout males, wild type females, and knockout females, and seeing how you might set up that kind of a design. There's also randomization with different factors of interest and more complicated ones as well. So you can have a look at these and see what might fit best with the experiment that you're trying to design, and then you can start off with a structure to alter it how you might want. So I'm just going to open up this example just to go through what it looks like initially. So when you open up, this is the actual example rather than the template, so it has some information inputted here, which you wouldn't have in the template, so you can add in your own details. But for this example, you can see on the left, we have what's called different nodes, and these are split into 3 sections. So we have our experiment, which is up here in the left. We have our practical steps, which is the sort of middle chunk here. And then we also have our analysis nodes, which are over on the right here. And this is basically encompassing the whole of our experiment, as you might expect. So the experiment nodes are summaries of the whole experiment overall. The practical steps is what we're actually doing. And then obviously the analysis is the analysis. I think something that's also useful with this tool is it has lots of help buttons. So if you're not quite sure what something is, you can press the question mark and it will come up with a help window on the right. So you can see here the experiment node is used to provide details about the null and the alternative hypotheses and the effect of interest. And every diagram should contain 1 experimental node. And so what this means is that you need to have a different diagram for every independent experiment that you're having. You don't want to put all of your experiments for one big chunk of work onto one diagram. You want to split it up. And that means that it can provide feedback and give you suggestions on how to improve if you just have one experiment in there. So the other thing you can do is go to the help section over here. If you're not looking for anything specifically down this side, and you can see it will give you information on getting started, creating a diagram, and give you some tips and hints over here, which is really useful when you're sort of getting started with this. So to go through a few details of this, or how to sort of read and navigate this diagram, you can see, as I said, the top left is overall experimental details. So you'll see this is the name of the experiment, effect of drug A on plasma glucose levels. If you click on this button at the bottom right, it will bring up more information, which has been filled in to some extent because it's the example. Obviously, if you're starting from scratch, it won't be, which I'll show you in a few minutes. So you can see it tells us, it has stars next to pieces of information that are crucial for the internal validity of the experiment, as it says. So these are things that we want to decide on before we've done our experiment and make sure that we've considered when we're designing it. And it also has these pieces of information with 2 stars, which are things that you can complete after you've completed your experiment and you have, for example, a link to the data that you've produced. You then link your experiment to your animal characteristics. So these are the animals that will be used in your experiment. And if we open up the properties here, you can see this is where we put information about the type of animals that we're using, so we can select a species. We can put in strain information, sex information, so whether they're male or female, or immature form, so you can't tell yet. You can also put in a whole load of other information like the genetic background, number of animals in cage, or anything else that is useful for your experiment here. So that's the sort of general overall. We also have under the experiment nose the experimental units, which is what I described briefly in the presentation. So in this case, this is mice, and the experimental unit is the animal. There are drops downs, so you can see it could also be cage or litter or whatever else you might want here. This experimental unit is linked to this blue square here, which is our group. So these are groups of animals that we use in the practical steps of our experiment. So the first group is always going to be our whole pool of animals that we have. So you can see we can say the total numbers that we need in our experiment with some justification for this. And I'll come back again to how we can use this tool to calculate our power. And also the role in experiments. So in this case, this is the total pool, but we also have options to show if it's a test or comparator group here as well. And then from this initial pool, we see that these will be subjected to some allocation. So we want to separate this into our two groups. And in this case, it's using complete randomization. So you're just going to completely randomize into group 1 or group 2, which are then subjected to some, in this case, pharmacological intervention, but it could be any kind of intervention down here. So if you're doing surgery, you can add in a surgical intervention instead, or whatever it is that your experiment is looking at. And then you take some measurement. So in this case, they're measuring plasma glucose. And again, it has some more information. You can see here, it has information on blinding. So how we're going to blind them. You can see it gives suggestions of how we might go about blinding them. So whether they're carried out by someone else who isn't fully aware of the experiment or doesn't know which groups are more likely to be higher or lower, for example, whether animals are individually coded, so they're sort of coded behind the scenes, you don't know what each one's getting, or whatever other strategy you're using for your blinding there. And then from this, we get our outcome measure. So in this case, glucose concentration, which again, you can add in information. This time you can add in whether it's continuous or categorical. So continuous or discrete is generally a numeric value. So in this case, you're measuring glucose concentration. So that will be numeric. You can also have categorical, ordinal, nominal, or binary. So this is when you have a category for your group. So this, for example, could be sex or sort of low, medium, high of a particular measure. And then we take our outcome measure and we want to analyze it. So we use that as an input. And again, we can include information about our analysis. And we can also get suggestions for which analysis we want to use. The final sort of part here is about our variables of interest. So in this case, we don't have any nuisance variables, so I'll show you how to add those in a second. In this case, we just have independent variables. So in this case, it's either a vehicle or a control, a vehicle or a drug A, sorry. So if we open this up, we can see again, we have the option of whether it's categorical or continuous, and more information on randomization and repeated factors. And you link, for our independent variable of interest, you always link every possible category where it is a category variable. So in this case, we only have two, our vehicle and our drug. But if we had more than that, we would have however many categories we had within that. So that's the general structure. I thought it might be useful to sort of start from scratch to show how you can add these things in. So what I'm going to do now is actually go back to my dashboard. I won't save those and just create a new diagram. So obviously, usually to save time, you can just use those example diagrams, but just to show how you might build one from scratch, I will do that. So we first want to start off with our experiment node, and we want to add some information about that. So as an example, I'm going to say that we are interested in looking at if there's a difference in weight between a particular control and a treatment group, for example. A completely hypothetical experiment, but hopefully gives you an idea. So we're going to be looking at effects on weight of some particular intervention. So we can call it that. Our null hypothesis will be that there is no difference in weight. And our alternative hypothesis would be that there is a difference in weight. And again, you will make these nicer titles if you're doing this for a particular experiment, but hopefully this will give you an idea. So our effect of interest in this case would be change in weight. And again, if you're not sure what any of these mean, you can go to the question mark at the top, and it gives you some statements that might help you decide what to put in any of these. The next one we have is the effect size. So this will be the difference in weight that would be of interest to us. So we're going to say 5 grams for this example. And then our justification, which might be that we've had previous data that shows that that has a biological impact on the health of the animal, for example. So then the next thing I'm going to do is add in our animal characteristics for this. So I'm going to drag in over from here, just click and drag to get this box here. You'll see both of these are outlined in red, and that's because they're not connected to anything. So to connect these, we can again click on this and we'll get an arrow and we can just drag it across. And then we'll say that this experiment uses animals from our animals characteristics box here. So in this example, I'm going to say that we're going to use both male and female mice, and we're going to account for that in our design. We're not necessarily interested in it, but we're going to include it as a nuisance variable. So my first animal characteristic, I'm going to say these are our female mice. So I will put in that they are female here. And then I'm going to need another animal characteristics for my males, because that's a different group within my experiment. So what I'm going to do is just copy and paste this, and then double click, and if, oh, sorry, click on the bottom right. And then if I'd filled in all of the other information, that would also stay the same. But I'm just going to change this to be male mice this time. And again, create a link in here. The other way of doing this is when you click on a particular node, it will give you suggestions for other nodes that it could be connected to. So you can see for an experiment, it can only be connected to one other type of node, which is our animal characteristics. So that also gives you some hints of how you can approach that. So the next thing I'm going to do is add in my first group. So if we put this down here, this is going to be my pool group, which contains all of my animals. So I will name this my pool group. I don't know how many animals yet. We'll come back to that. And we can come back to experimental units and things, but I'm going to tell it that this is my pool group. So this is all of my animals. And if we remember, we also want to define our experimental units. So we can link these to our pool group, either by dragging it out from here or selecting the same color and shape from here. So you can see if we click on that, it will automatically link it as well. So in this case, our experimental units are going to be each individual mouse. And we can also say specifically that it's each animal. You can also play around with where you place things by just dragging and dropping, and you can make your arrows have sort of bends in them if you want to make sure that they are, your text is sort of more horizontal. So that's the initial setup for our pool group. We now want to allocate this to our two different groups. So we can see allocation is our blue circle here, or we can click it over here. We can see our pool group is subject to an allocation. So this time, because we've got both male and females, we're going to allocate them slightly differently to in the example. So our randomization strategy here is going to be instead randomization within blocks. And that's because we're going to block for our male and our female mice in this. We can say what's randomized. We're randomizing our experimental units to our treatment. And we can also say what randomization procedure we're going to use. So we're going to use the EDA spreadsheet, and I'll show you how to do that soon. And again, we're going to ask someone else to carry this out so that they are sort of blinded in this case. And then you can see this allocation box only has one option. So we're going to create a couple of new groups because we're allocating into our control or our treatment. And again, we can say the role in the experiment here is our control, and we can call group 1 our control, so we can keep a track of that. And here is our treatment, and we'll say it is our test group here. And then we want to add in our intervention. So let's say this is some pharmacological intervention. Then we add a separate one for our control and our test, because there are different groups. And then we want to create some kind of measure from this. So in this case, we're going to measure their weight because that's what we're interested in. So we can click to add a new measure. This time the measurement's going to be same for both. So we can just create one and then create a link from our test group here too, so that they both get this measurement. And again, we can say whether it's blinded or not, and how we're going to decide which order we're going to measure our groups in. So again, this can be randomized, for example. And we can say that the person measuring is going to be carried out by someone else, for example. So we've now got the majority of our experiment and the majority of our practical steps. So we want to add in our analysis at this point. So from our measurement, we're going to get some kind of outcome measure, which in this case is going to be weight. So we can add that in and create a linker here to say that this is coming from this measurement. And again, we can put in more information about our outcome measurements. So it suggests that we should say whether it's continuous or categorical. In this case, it's weight, so it's going to be continuous. And we can also say if it's a primary outcome measure. So if you're doing multiple measurements within this experiment, there might be one that answers your key question, but some others that mean that you can get more information. In this case, we're only doing one. So yes, that's going to be our primary outcome measure. And then we want to add in an analysis that we're going to do on this. So we're going to drag and drop that in here. And because we're doing it on our outcome measure, we again want to link these. You can see outcome measure only has two possible options. The bottom one is the analysis. The top one is a data transformation. So if you know, for example, that your data generally follows a log normal distribution, and usually you have to log transform your data to do your analysis on it, you can add these steps in specifically as well to really show the entire process, including the process in the analysis as well. So we're going to link these two. And you can see then this outcome is an output for our analysis. So now our analysis wants to take into account some measure of interest. So we need to add in our independent variable of interest so that we can say what we're actually using in our analysis. So if we drag that in here, in this case, our variable of interest is our two different interventions. So we can put that in here, which might be a specific drug, for example. And again, we have a control or a treatment, so that's going to be categorical. You can also say whether it's a repeated factor. So if you're doing multiple measures, you can add that in. In this case, we're not. And again, this isn't connected to anything where it wants to be. So we're going to add in our two different groups, which is our control and our treatment. And then we're going to link this to our analysis as well, because that is our factor of interest for our analysis here. So that seems fairly complete, potentially. This sort of follows the same steps as we had in our example. So if we're fairly happy with what we've done, but we're not sure if we might have missed some things, we can go up to the top here and go to Tools, and there's an option to critique our diagram. So I'm going to click on that, and that's going to take a minute because it runs through everything. So I'll keep talking while it's running. But basically, it's going to give us suggestions for how we can improve in the form of things that we really need to look at. So we get some warnings, some suggestions, and sometimes there's also just some advice for things that aren't necessarily incorrect, but you perhaps haven't thought about. So that's finished now. You can see we've got 6 errors. So these are things that we definitely need to correct. We've got 11 warnings and 1 advice. So perhaps we've not quite finished on this yet. So the key things that we definitely want to address are the exclamation marks, as you might expect, in red. So let's have a look at a few of these. So we can click on them and it will open up more information. So for our allocation strategy, we've randomized within blocks, but we haven't told it what our blocking factor is yet. So we need to add in our sex as a blocking factor to this. So to do that, we can drag in a nuisance variable over here because it's not a factor that we're necessarily interested in, but we want to control for. So this looks very similar to our variable of interest, and similarly to this, we need to add in our 2 categories here. So these are going to be, these are going to be our males for our females to keep them in the same order as above. And these are going to be our males. And our nuisance variable in this case is sex. Again, this is categorical. A big reason why it asks for this is it will then help when it suggests an analysis, as we'll see in a second. And how is this variable accounted for? It's going to be blocked. And then what we want to do is assign it to our allocation here. And we get a red arrow because it needs a bit more information about what's going on. So if we click on it and click on the little red spanner here, we can see it's a blocking factor for our allocation. So what this means is it will take your males and your females separately, and it will randomly allocate to group 1 or group 2 within your males and within your females. Okay, so now let's look at our other exclamation marks. So we can see here that our groups are not differentiated. It can't see a difference between at least 2 of the groups. So the reason for this is that we haven't connected our variable of interest to our groups down here. So to do that, we can just copy and paste to get another sort of similar block, and then we can place that inside of our groups. And that will tell us specifically that group 1 is our control group, and that group 2 is our treatment group down here. And these exclamation marks up here is that it isn't tagged to the main part of the diagram. So that's the same issue as the one that we've just changed. Also, just to say, I did that quickly, but there's a button in the top left that means you can resize any of these as well to make your diagram look nice. We also have an exclamation mark over here, so let's see what this is. It says the outcome measure is not defined. So we haven't renamed this label. It's still just called outcome measure 1, which the tool doesn't like. It likes you to define what it actually is. So we're going to call this weight, and then it'll be happier. So the next thing we want to look at are all of our warnings. So you can see a lot of these are going to be about sample size and effect sizes, because we haven't given much information on that yet. There are also things about inclusion and exclusion criteria that I haven't gone into, but it does like you to include that at the start. And other sources of variability that might be an issue. In this one, again, this is mostly about sample size, which we haven't defined yet. So we'll look at how we do that in a second. Again, these two are both just about sample size. The analysis, we didn't put too much information in. So it wants to know about blinding status. It wants to know what method of analysis we're going to use and any assumptions that we're going to make. And again, it's asking for inclusion and exclusion criteria. So we can add in those things. We can also get help on our power calculations and analysis options from the tool itself. So if we go back to the tools window, you can see the option for sample size calculation and analysis suggestion. So we're going to do the analysis suggestion first, because we need to know what analysis we're doing before we do our sample size calculation. So I'll click on that. And again, it will take a second to figure this out. So I'll go back to the tools and just show you can also get your randomization sequence from here, and output an experimental design report, which I'll show you an example of in a second, and also a read-only diagram. So if you're working on experiments with many other people, as most people do, you can export a read-only diagram and share that so that other people can download and play around with your diagram as well. So it's finished running the analysis suggestion. We don't have any warnings and we have one suggestion. So this comes up to our analysis node here. And it gives us a summary based on our diagram, what it thinks that we are looking at, to make sure that it has correctly assumed things and that you haven't put anything in incorrectly. So we can see this analysis should make a comparison between two groups, which is what we're doing, including a single continuous outcome measure, which is our weight, and two groups, which are our categorical factor of interest. And there's also some blocking. So, might be because I didn't complete after the blocking, actually. So maybe I need to do something different here. We can see if we just had two groups, then we would be doing an unpaired t-test and the Mann-Whitney test. But this isn't taking into account our blocking by sex here. So there must be something else that either we're missing or because we didn't run our critique again. So we're going to go back to our tools and critique again. And we'll see where maybe we've gone wrong with this, or if it just needs to update. So to go back to some more things while that's running, you can see anything that you can do sort of using buttons. You can also do up here, so you can delete any nodes, copy and paste them. You can also group things so that when you're moving them around, you can move them around all at the same time. And you can zoom in and out and change your size of your diagram and everything to make it look nicer. You can also share, again, your diagram so that other people can access it and make changes to it. And you can export it to save it in your own sort of drives as well. You can also save it as either an experiment, or you can save your own templates as well. So if there's a particular experiment that you know you're going to be running multiple times, you can save a template for it and then reload the template each time, so you don't have to create the same structure all of the time. You can just change the factors within it. So our critique has finished running. We can see this time we don't have any errors, so that's good. That's an improvement, but we still have 12 warnings and 1 advice. So we'll go okay on that. And we can have a look at some of our warnings. Some of these are going to be the same as what we had before. So we can see blocking factor in the randomization should be taken into account in the analysis. So this is perhaps where we went wrong before and why it wasn't included, because we haven't told the analysis that we're interested in this as a blocking factor. So to do that, we can either just connect this node all the way over here to our analysis node, or again, we can, press one button, copy and paste this so that we don't have arrows, so that we don't have arrows going all the way across our diagram. And then we can connect this to our analysis here and tell it that it's a blocking factor. And so now if we rerun our analysis suggestion, it should take into account also the blocking factor and tell us that we want to do a one-way ANOVA with blocking. I won't run that because it takes a couple minutes to run again. So if we know that, or if we actually just want to do a two-group comparison as well, we can calculate our simple sample size power calculations within the tool. So using this option here, it will come up with these options. We can only do t-tests on here. So if you want to do anything more complicated than that, you will have to use different software and or come and talk to me. So you can see it's super simple. All we have to put in is an effect size. So if you remember when we created this node, we said an effect size. So a change in weight of 5 grams would be of interest. So we can add that in. And then we have to also add in information about the variability that we expect. So say we expect a standard deviation of 3, we'd probably get this from like past experiments, for example. And then we have our significance level. So this is our significance threshold of usually 0.05 and a power of usually 0.8. And we can calculate our N per group. And you can see based on a difference of 5 grams and a variability of 3 grams, we would need 7 mice per group. So we could take that and add that in to here. So we would need a total number of 14 animals, which is the same as our experimental units. And we can add in a justification saying that we did a power calculation based on a difference of 5 grams, a standard deviation of 3 grams, and a power of 80%. And we can also add in for each of these to say that we need 7 animals and 7 experimental units, etc., etc. So that has now given us our analysis suggestion and our sample size calculation. So we are pretty much done here. Obviously, we can tidy it up and make it look nicer, but for the sake of this course, I won't worry too much about doing that. So now we want to share this with someone else, and we want to create a nice report that we can include in our grant application or our project license or anything like that. So to do that, again, we use the tools window or the tools drop down, and we select either design report, which will create a PDF, or a read-only diagram. But again, I won't run these because they take a couple of minutes. So I will just bring over an example that I made earlier of a report so you can have a look at that. So bring this over here. So this is the kind of report that it produces. You can see it gives you a title of your report. This will be the title of the experiment that you did. So in this case, it's just a copy of the example that I showed you before. It shows you the date that you did it, and prints out your null and alternative hypotheses, and it gives you all of the information that you need for the arrive checklist that I mentioned earlier. So again, this is really helpful. It takes a lot of work up front, but it's a really nice and easy way to collate all of this information that you will need at some point, and throughout, both when you're planning, designing, running, and reporting your experiment. It means everything's all in one place. You can see in this one there's a lot of information not provided, but obviously if you've done a better job of filling everything in, then it will have all of your inclusion/exclusion criteria and things like that. It includes your randomization and your blinding strategies, and your outcome measures. And it also tells you the statistical methods that you're going to use in your analysis. Again, it includes all of your information on the experimental animals that you'll use. So if you've included information on the species and strain and things, that will all be included in here. And then it gives you your nice diagram of your experiment, so you can see the whole thing and see the sort of steps of the practical implementation of both your experiment and your analysis. The final page is a summary of the feedback. So these are the critiques that you haven't specifically answered in your diagram. So this can be really useful for people who are reviewing your experiment. They can look here and see that you haven't, in this example, said anything about the sex of the animals, and that maybe they should go back to you and say, well, have you thought about this? Why haven't you included this in your design, etc., etc. And it also gives the advice for the primary analysis. So this is the suggested method that they think that you should use to analyze your data, which is likely going to be the same as your statistical analysis method up here. So that was a very quick overview. I'm going to go back to my slides to just quickly, oh no, there was one more thing I was going to show you, which was the randomization. So if you select to create a randomization sequence here, it will let you input an email address. So it doesn't have to come to you. It can go to someone who is then helping to blind your whole experiment so that you are not aware of what groups different mice fall into in this case. And they will receive an email, which will have an attachment of a spreadsheet that looks very much like this one I made earlier. So in this example, we had 10 animals in both male and female groups. So you'll see it will have your 10 animals. It gives you a space to add in your unique identifier, and it gives them a random allocation for your males and your females in this instance. And it also gives instructions for how to use this as well. So it saves you having to go into Excel and create random numbers and things like that. It will just generate it all for your different groups for you, which can also be super helpful. Okay. So I will actually go back to my slides this time. Here we go. So that was a very quick overview, but hopefully it gives you an idea of some of the things that the EDA can do and how it can help. So it basically helps to design robust and reliable experiments. It makes suggestions and gives you prompts to consider things like experimental units, your blinding, your randomization, any nuisance variables. It helps you decide on statistical analyses and sample sizes as well. It lets you produce nice diagrams and your reports so that you can share how you've designed your experiments, which makes it easier to get critiques back as well on how to improve from other people. And you can also include these in grant applications or alongside project licenses, and all the information is there in one place. You can, instead of using a blank starting point, you can use the templates as a starting point to give an initial structure and just edit those. And you can also save templates so you can reuse them at a later date. If you get stuck, there's always the help options that are really useful to remember. And you can at any point critique your diagram to see any things that you might have missed while building it. So my final slide here is just about some further resources. I can, if you're at the Veblen Institute, provide advice, help to produce diagrams or templates for experiments. This is something that I'm hoping I can help to sort of implement more within the Institute. So I'm really happy to help with this in any way I can. The EDA website is really useful, and they also include further demo videos if you want to take a look at those. The NC3R's website has a load of resources on experimental design and beyond. There's also the ARRIVE guidelines that I mentioned, and their study plan, and the PREPARE guidelines, which I've included all the links for there. And finally, something that I found that I thought might be useful more generally for experimental design is there's some experimental design training that was developed by the University of Nottingham and an organisation called FRAME, which is the Fund for Replacement of Animals in Medical Experiments. And this goes through a lot of the considerations for experimental design. It's just an online sort of self-directed training, so you can go through it at your own pace and dip in and out and look at anything that might be helpful for you. So I hope that was useful, and thank you for listening.